start. So this is the things that we're going to cover today, starting with the internal bacterial structure and function. Okay. So it doesn't work. Okay. Starting with this, which is basically just the cytoplasm. So what is the cytoplasm? It's present in every cell. It's made up of mainly water, 70 to 80% water. And an important function of cytoplasm is that it contains all the molecules, and therefore it's the storage of energy of the bacteria. So it's the energy storage of a bacteria. And cytoplasm, also very importantly, is the site of replication. So if you're asked what is the site of replication in a bacteria, then you, instead of all the other organelles, you can answer cytoplasm. And it's also where there's going to be growth and metabolism, which makes sense. So that is basically the cytoplasm. Now we add this structure. So what is this? I think this is. Okay, yeah. So this is the chromosome. And what is the chromosome? It's basically just the DNA of the bacteria, and it's only one. The bacteria has only one chromosome. And since prokaryotes, which, for example, bacteria, they're prokaryotes, they do not have membrane-bound organelles. So this chromosome is just lying freely in the cytoplasm. There is no membrane surrounding it, and therefore we do not call it a nucleus. Instead, it's a nucleoid. So it's like a nucleus, but it's not a nucleus because it's not bound by a membrane. It lies freely in the cytoplasm, and it's a double-stranded DNA, like most DNA. Most of the times it's circular, like circular DNA, but sometimes it can be linear. So if you were asked, mostly it's circular. And our DNA, it has histones. So histones are these proteins that the DNA wraps around. The chromosome or the DNA in a bacteria does not have histones, and therefore they call it naked DNA. Okay? So that is about the chromosome. Now this little structure appears. What do you think this is? Okay, correct. So this is the plasmid. And if you guys remember, what is one really important function of a plasmid? What do you think is like an important function? 2-1. Okay, so we'll start with what a plasmid is. So plasmid, since it's DNA, it's also DNA, it's nucleic acid, but it's not part of the chromosome. So if it's not part of the chromosome, then we're going to call it an extra chromosomal DNA, okay? And since it's extra, it's not present in all bacteria. So not all bacteria have it. And having it is good for the bacteria. It provides a selective advantage because it contains genes for antibiotic resistance. So having the plasmids is going to, it might have the antibiotic resistance, and it's going to help the bacteria, okay? And if a bacteria has it, then it's called F positive, F standing for fertility. So the bacteria is fertile, meaning that it can transfer this plasmid to another bacteria through a process called conjugation. So plasmids in fertile bacteria can transfer to another bacteria, and this process is called conjugation, and we're going to talk more about it later. Okay, so is that clear, everything until now? Yes. I'll move on to the next slide. So here we have these bodies. What are these called? What do you think these are? Ribosomes, perfect. And what do ribosomes help in? Protein synthesis, perfect. So what are ribosomes? Ribosomes are just these small dots, I would say, that are made up of RNA, ribosomal RNA, and proteins. So they themselves are made up of RNA and proteins, okay? And in a prokaryote, the ribosome, the size, and this is important to remember, is that there are 70 S ribosomes, okay? So in prokaryotes, in bacteria, the ribosomes are 70 S, okay? Made up of two subunits, which are 50 S and 30 S subunits. As you can see, for example, this is the structure of a ribosome. We have a bigger subunit, which is the 50 S, and a smaller subunit, which is the 30 S, okay? The ribosomes in a eukaryote, what is their size? Are they also 70S, or what size are they? If you guys know. 80S, yes, perfect. So in a eukaryote, it's 80S. In a prokaryote, they are 70S ribosomes, okay? And ribosomes are the site of protein synthesis. And why is this important? Because there are some antibiotics and antibacterials that we can use specifically to target the protein synthesis and target the ribosomes. And these antibiotics, and you'll have to know them for the exams, some of them inhibit the 30S subunits, for example, aminoglycosides, and some of them are going to inhibit the 50S subunits, okay? So that is for the ribosomes. Moving on to these things, which are like small bodies that vary in size. These are inclusion and granules, okay? So inclusions and granules are small bodies in the cytoplasm, and they vary in size and number. They're not always present in bacteria, okay? Sometimes they're there, sometimes they're not. What makes them appear in a bacteria? Usually, if there's depletion of nutrition, there's a harsh environment, and the bacteria needs to store some nutrients and store some, it's basically like a storage unit that appears when there is a lack of nutrition, okay? And staining bacteria sometimes and seeing inclusions and granules can help identify some bacteria, okay? So as we said, it helps their storage sites that can help when the nutrients are depleted from the environment. And usually when the nutrition is back, they can disappear. So they're not always there. They're temporary storage organs. Energy storage is mainly in the cytoplasm, okay? And then we have mesosomes. So what is mesosome? If you can see here, there's this like invagination or folding of the cell membrane, and it's usually like vesicular or laminated or tubular. So there's going to be like tubules and stuff inside. And why are mesosomes important? Because they mainly help with the respiration. We know that, as we said, that prokaryotes, they do not have any membrane-bound organelles. So that means that they don't have a mitochondria to help them with the respiration. So mesosomes can help with the respirations, but they're not always there. Again, just like inclusion granules, they're temporary and they appear more in gram-positive bacteria. What are gram-positive bacteria? We're going to see in a while. I'm going to explain, but mesosomes are mainly in gram-positive bacteria, okay? And sometimes they may play a role in replication, okay? So that is for mesosomes. So now I want you guys to match. You can either say in the chat or unmute yourself these to what structure they are, okay? Starting with antibiotic resistance. Which one helps in antibiotic resistance? Plasmid. Okay, yes, correct. Okay, so antibiotic resistance, it helps with plasmid. Okay. Which organelle has two subunits? Ribosomes, perfect, yeah. So ribosomes have two subunits. What about which organelle helps in energy storage? Not temporarily, but usually. Not the temporary one. Yes, granules also help. They included in granules. Yes, cytoplasm. So cytoplasm is a site of energy storage. Okay. Naked DNA. Chromosome. Yeah. Which organelle temporary organelle helps in respiration? Yes, mesosomes. Okay, perfect. And they're temporary. Site of protein synthesis. Which organelle? Ribosomes. No, cytoplasm is where everything is there. So the ribosomes are in the cytoplasm, but ribosomes are the ones that will do the protein synthesis. So nutrition related, I mean, for example, it appears and disappears based on the nutritional status or environmental status, which organelles? Yes, inclusions and granules. So when there is harsh environment, they will appear to store the nutrients, and then when nutrient comes, they're going to disappear. Which organelle is transferred through conjugation or can be transferred through conjugation? Plasmids, perfect. And which ones can be stained and help to identify the bacteria? Sometimes. Yes, the inclusions and the granules. Which organelle is more in gram-positive bacteria? Mesosomes. And which one is a nucleoid? Can also be called a nucleoid. Chromosome, yeah. So it's not a nucleus because there's no membrane-bound organelles. Which one is the site of replication? Which organelle is the site of replication? Cytoplasm, perfect. And the last thing, may play a role in replication. Mesosomes, perfect. Okay, so this is for the internal bacterial structure and function. You reached a checkpoint. If you guys have any questions, you can ask me now. You can unmute or type it in the chat. Otherwise, we can move on. Everything clear? Okay. Okay, so we'll move on. So now we're going to look at the external structure, which is the cell envelope. Okay. So first we have the cell membrane. So what is the cell membrane? It's a phospholipid bilayer, as you can see, one layer of phospholipids, the second layer of phospholipids, just like our cell membranes, okay? However, the difference is that they do not have sterols. For example, our cell membranes, they have cholesterol, right? But the bacteria, they do not have those sterols, except, for example, some bacteria like Mycoplasma. Mycoplasma has sterols. Otherwise, they do not have sterols, okay? And just like our cell membrane, they're going to control interaction between inside the cell, outside the cell. They have the transport of molecules. But what's new is that cell membranes help in respiration. What other organelle we said helps in respiration? Mesosomes. Yes, perfect. So, and that makes sense because mesosomes are invaginations of the cell membrane. So if the cell membrane helps in respiration, then mesosomes can also help in the respirations, right? And why does the cell membrane here take over the respiration role? Because there is no mitochondria, okay? And why is there no mitochondria? Because we don't have any membrane-bound organelles. So in this case, we're going to have the cell membrane take on the role of respiration, okay? Another thing is that it helps in biosynthesis and synthesizing what? Synthesizing the cell wall of the bacteria, okay? And very important, cell membrane is the site of septum formation. So in the cell division, where does the septum formation happen? In the cell membrane, okay? Then we have this structure, which is the cell wall. okay? And the cell wall is made up of peptidoglycans, okay? And the peptidoglycans, from the name, peptidopeptides, these are proteins, right? And glycans are sugars. So the cell wall is made up of peptidoglycans and sugars. And since it's a rigid structure, it's going to give the cell its shape, whether it's bacilli or cocci, it's going to give the cell its shape. And very important is that it's going to help in resisting osmotic pressure. And why is this important? So say, for example, you give an antibiotic that weakens the cell wall. What happens is that the bacteria, for example, now is no longer able to resist the osmotic pressure, and therefore it can actually burst. So this is one of the mechanisms of actions of the antibiotics, right? So important, resist osmotic pressure and gives the cell shape. Another thing is that it's going to protect the bacteria, which makes sense. It's a wall around the bacteria. And another thing is that it contains virulence factors. So what are virulence factors? Do you guys know what virulence factors are? Yeah, they give bacteria its pathogenicity, which means that basically it's how virulence factor is the things that are going to make the bacteria more infective or give it an advantage in being more disease causing, right? So that is what virulence factor is, and cell walls contain a lot of virulence factors. And cell walls can also play a role in synthesizing their own self, the cell wall. Okay, and also against cell division, which makes sense because when you divide, the new bacteria has to also have their cell wall. Okay, and important or something that's beneficial to us is that sometimes the cell walls also contain antigens, and these antigens the body can form antibodies against, and we're able to, you know, kind of have immunity against this bacteria. Okay, so that is for the cell wall. And another very important thing about the cell wall is that it helps us differentiate between gram-positive and gram-negative bacteria. Okay, so this is a cell envelope of a gram-positive bacteria, and this is the exterior of a gram-negative bacteria. What are some differences that you can notice? Which slide? Sorry, this one? Yes, okay. You mean the cell membrane or the cell wall or both? Mm-hmm. Okay. So the cell membrane, as we can see, let me remove it. Okay. So the cell membrane, it's made up of phospholipid bilayers. Okay. So there are two layers of phospholipid. This is the first layer, and this is the second layer. Okay. So it's a phospholipid bilayer. And it doesn't have any sterols. And as we said that we, as eukaryotic cells, we have sterols like cholesterol in our cell membrane. However, in a bacterial cell, they do not have sterols. For example, mycoplasma. Mycoplasma has no, they don't have sterols, most of bacteria, except mycoplasma, they have sterols. This is the exception. Most of the time, they don't have sterols, okay? And just like our cells, they're going to be involved in transport of molecules and permeability, basically. However, the additional thing is that they help in respiration. And why is the cell membrane taking over the role of respiration? It's because the bacteria, they don't have mitochondria. And why don't they have mitochondria? Because they don't have membrane-bound organelles in general, okay? And then also, the cell membrane is where the septum formation happens. So the septum formation happens in the cell membrane. And it also helps in cell wall synthesis. For the cell wall, we said it's made up of peptidoglycan, peptides, which are proteins, and glycans, which are sugars. And since it's a rigid structure, it provides the cell shape and also resists against osmotic pressure. So if there's pressure of like an osmotic pressure on the cell wall, it's not going to burst. But there are some antibiotics, if we weaken the cell wall, then the cell is going to lyse because it can no longer resist osmotic pressure. Therefore, it's also a protective mechanism of the cell wall, of the bacteria. It gives it protection, and virulence factors, which we said are the things that are going to help the bacteria be more disease-causing and more infective. And these virulence factors, and then there's also antigens on the cell wall. And antigens, what are antigens important for? Is that the cells of our body, they can recognize these antigens and then form antibodies against these antigens, and we're able to kill those cells, basically. So it's helpful for us being antigenic. Okay, is that clear? Okay, move on. So we said, no problem. So we said that also the cell wall or the cell envelope structure helps us to differentiate between gram-positive and gram-negative bacteria. So can you guys tell me what are some of the differences that you can see between a gram-positive cell wall and a gram-negative cell wall? Or a cell envelope, I would say. You can unmute yourself if you find that easier. Thicker cell wall in gram-positive, that makes sense. Look at how much thicker it is. Usually we have like 50 to 100 layers of peptidoglycan in a gram-positive, but in a gram-negative, it's like 2 layers or so. What else can you see is the difference? Outer membrane in gram-negative, yes. So in gram-negative, although it has less cell wall, it has something to compensate for it, which is an outer membrane, right? Okay, yeah, perfect. Periplasmic space in gram-negative, true. So here we have a periplasmic space, but it's very, very small that you consider it like non-existent. However, in a gram-negative bacteria, you can see that all of this is the periplasm, so it's much bigger. So you do consider that periplasm is in the gram-negative. Okay. So let's look at some of the differences. So in gram-positive, what is gram in general? Gram it's a stain. So based on the staining, bacteria can be gram-positive or gram-negative, okay? So in a gram-positive bacteria, the stain would look purple. So the bacteria will look purple when you apply gram stain to them, okay? And gram-positive, we said, has no outer membrane. It has a thick peptidoglycan layer, if you can remember from before. And we said that the periplasmic space is kind of non-existent. It's very small. You consider it not there. And the virulence factor of a gram-positive bacteria include, if you go back, they include these two things, which are teichoic acid and lipoteichoic acid. Teichoic acid is a shorter name than lipoteichoic acid, so it's shorter. And lipoteichoic acid is much longer and it reaches the cell membrane, okay? So we said the virulence factor includes teichoic acid, which is short, only within the cell wall. And lipoteichoic acid, longer name, LL, it's long and it's attached to the cell membrane, okay? And what are those acids? They are polymers of phosphate and ribitol or glycerol. So they're polymers of phosphate and ribitol or glycerol, and they contribute to the negative charge of the bacteria. So if you're asked for a gram-positive bacteria, what provides the negative charge? You can say teichoic acid or lipoteichoic acid, okay? And they can bind calcium, which makes sense. If they're negative, they can bind positive molecules, they attract. And they also have antigenic properties, which we said against antigens, what forms? Antibodies, okay? So what about gram-negative? Gram-negative, if gram-positive is purple on gram stain, gram-negative is pink on gram stain. If gram-positive has no outer membrane, then gram-negative, if gram-positive has no outer membrane, then gram-negative has an outer membrane. If gram-positive has thick peptidoglycan, then gram-negative has thin peptidoglycan. If gram-positive has kind of no periplasmic space, then gram-negative has periplasmic space. If these are like the main things that are opposites to each other, but now let's look at the virulence factors of gram-negative bacteria. So another thing is gram-negative contains porins, but also what are the virulence factor? The virulence factor is mainly one thing in the gram-negative bacteria that's called a lipopolysaccharide. Yes, LPS. Perfect. So lipolipid polysaccharide, so sugar. So it's composed of lipids. So we know for sure that it has a lipid and it has a sugar. And we're going to look more at its structure later. But basically, what do they have? They have O antigens or O side chains. And they're antigens. And if we have antigens, then our immune system can recognize them, have an antibody response against them, and we become immune, right? Another thing is the core sugar. So it's saccharide. So the core sugar is what gives the gram-negative bacteria its negative charge. So gram-positive, the negative charge is from the teichoic lipotechoic acid. Gram-negative is from the core sugar of the LPS, okay? And then we have lipid A. Lipid A, so all antigens, they are going to have the antibody response. Lipid A is going to give the inflammatory response. It's going to activate like cytokines and stuff that's going to cause inflammation and fever and sometimes septic shock. So lipid A is the kind of the bad component of the gram-negative bacteria, okay? So let's look at the periplasmic space and then we'll go back to the other organs. We're going to look at each one of them separately. So periplasmic space, we said, is going to be the space between, So it's the space between the external surface of the cell membrane and the internal surface of the outer membrane, okay? And we said it's mainly present in gram-negative bacteria, okay? So what does it contain? What's inside it? What can you see? You can see that there's the peptidoglycan. So it has the peptidoglycan inside it. And also itself is kind of a gel-like layer that has proteins and other molecules and stuff like that, okay? It contains sugars, it contains metabolites and stuff like that. It also, very importantly, contains enzymes. And these enzymes can be lytic enzymes, like what lytic means it breaks down. So it can be enzymes that break down lipid, lipases, enzymes that break down protein, proteases, enzymes that remove phosphate, phosphatases, and stuff like that. Or detoxifying enzymes, which is, I would consider, the most important function of the periplasmic space, is that it contains detoxifying enzymes like beta-lactamases. What are beta-lactams? Beta-lactams are antibiotics, like penicillins and stuff, that are toxic to the bacteria, right? So if you have enzymes that can detoxify them, beta-lactamases, it's going to make the bacteria resistant to these beta-lactams, right? And this is a question outside from this lecture, but if you have beta-lactamases, what can you do, or what did you study? You can do to help the antibiotics, the beta-lactams, to still be able to kill these bacteria, even if they have beta-lactamases. What do you add to a beta-lactam to protect against the beta-lactamases? Yeah, exactly, beta-lactamase inhibitor. So for example, you have amoxicillin, which is a beta-lactam. You add to it clavulanic acid, which is a beta-lactamase inhibitor, and you're going to be able to kind of overcome this method of antibiotic resistance, okay? Now we're going to talk about the outer membrane. So what is the outer membrane? We said, again, it's part of the gram-negative bacteria, and it's another lipid bilayer, but it's not symmetric. So this is symmetric, both are phospholipids. This is not very symmetric. Like this is phospholipid, but here we have some lipopolysaccharide and stuff like that, okay? So it's an asymmetric lipid bilayer made up of gram-negative bacteria. It's only present in gram-negative bacteria. and it's made up of phospholipids, liposaccharides, and it has membrane proteins embedded in it, okay? And if you can see here, the structure of the lipopolysaccharide, these are the lipopolysaccharides, as you can see. And it has at the bottom of it lipid A, which we said, what's the function of lipid A? Yeah, it's a virulence factor, but what does it do? It is an endotoxin. Yeah, inflammation. So it causes inflammation. And then we have the core polysaccharide, which we said is the negative charge. And then the repeating oligosaccharides, or the O side chain, O antigens, what do they do? What do they help in? Antigen, yeah, recognition, which will help us to produce what? Antibodies, perfect, okay? So we form antibodies against these O side chains or O antigens, okay? And then the membrane proteins, you can see one of them is a porin, okay? And we're going to talk more about the porins in a while. So basically, this outer membrane is connected to the peptidoglycan through something called brown lipoproteins or lipoproteins. And these lipoproteins, what's their function other than connecting? They provide the cell with its integrity. So the cell integrity is what is the function of what? Of the brown lipoproteins. Cell integrity, brown lipoproteins, okay? The outer membrane is, again, since it's the outermost, it's going to be the permeability barrier, and it's going to help in selective things that are going in, whether they're hydrophobic or hydrophilic, large or small molecules. And usually these smaller molecules, hydrophilic molecules, are going to pass through the porins, which we're going to talk about right now, okay? So the porins, which are these things, they are basically the proteins embedded in the outer membrane, not the cell membrane, in the outer membrane, again, present in gram-negative bacteria. And what's their function? The transport of small hydrophilic molecules inside or outside the cell. And porins have a very important function. In what? What do you guys think? Do you guys remember an important function of porins? It's kind of considered like a virulence factor. So porins, they mediate antibiotic resistance, okay? Kind of. So like porins basically sometimes, for example, there are some antibiotics that can only go in through the porins. So that's the passage for them. And if a bacteria then decides to, for example, downregulate the porins, there's going to be less antibiotics that can go in, and you're going to have antibiotic resistance. So porins, they help in the mediation of antibiotic resistance, okay? Then we have glycocalyx. So glyco means sugar. So it's something sugary, and calyx means covering. So it's a covering made up of sugar, or a coat made up of sugar. So what is a glycocalyx? It's an adhering layer of polysaccharides, which are sugars, that adheres to the bacteria. Sometimes it can be amino acids, made up of amino acids like anthrax bacilli, so AA, amino acid anthrax. But most of the time, the glycocalyx is made up of polysaccharides, okay? And in the gram-positive bacteria, it's going to be outside the cell wall. In a gram-negative bacteria, it's going to be outside the outer membrane, which makes sense because the outermost layer of gram-positive is cell wall. The outermost membrane of a gram-negative is an outer membrane, okay? If the glycocalyx is organized neatly in a very neat layer, this is called a capsule, okay? So for example here, this is a capsule, a glycocalyx that's organized neatly. However, if it's diffusely or loose organization, slime layer, this is called a biofilm. For example, this bacteria, this glycocalyx is not neatly organized, so this is a biofilm or a slime layer, And what is the importance of this glycocalyx? It mainly acts as a virulence factor, okay? So having a glycocalyx, a capsule, or a biofilm is a virulence factor for a bacteria. How? So these glycocalyxes help in adherence and colonization, okay? So if I were to give you an example, there's this bacteria that, for example, if you insert, if they tell you in the question that a patient had a catheter or an intubation, and then he got an infection, usually this bacteria is going to stick to this foreign object, and it's going to form this biofilm, for example, that protects it. So it adheres to the foreign object and colonizes it. And then again, this biofilm is actually going to protect the bacteria from being killed by the immune system. What bacteria am I talking about, if you guys know? Like an example of a bacteria with a biofilm. Staph epidermidis, perfect. So a very hallmark thing of staph epidermidis, if you see in the question any catheter or foreign object insertion, think about staph epidermidis, okay? So staph epidermidis has this biofilm. So they help in adherence and colonization. They protect against phagocytosis. So usually capsules is not, since they're kind of very organized and the bacteria already has it, it's mainly the capsule is protect against phagocytosis, while the biofilm is produced by the bacteria later, and it causes a covering and makes it stick and stuff like that. That's usually the one for the binds to the foreign objects and stuff like that. So I would say that this mainly works for a biofilm, and for the capsule, it's mainly against the phagocytosis. Okay, no problem. And also very important is that the capsule is actually antigenic. Again, what does antigenic mean? Antigenic, basically, the immune system can recognize it and form antibodies against it. So why is this important? Number one, like we said, we can form antibodies against this antigen and help our system. Another thing that's very important is that since it's antigenic and you can actually identify the bacteria with it, specific bacteria. So through serologic testing, you can identify what the bacteria is through these antigens. Another thing is that since we know the antigens and we know that we can form antibodies against these antigens, we can replicate these antigens and make a vaccine that we can vaccinate people with and provide immunity, right? So the immunity is going to form against these antigens in the polysaccharide layer or the capsule layer of the bacteria, okay? So what are some examples of bacteria with a capsule, or for example, Klebsiella pneumonia, which I heard you guys didn't really take, Neisseria meningitidis, strep pneumonia, and Haemophilus influenza. And what is what is common between all three of these bacteria, all these three encapsulated bacteria, is that they all cause meningitis. So another thing is that Dr. Atif used to tell us, if a bacteria is capsulated, know that it can cause meningitis. It can cross the blood-brain barrier and cause meningitis, okay? Capsule equals meningitis. And so we also have vaccines made up of the polysaccharide layer, so made up of the capsules of Neisseria meningitidis, strep pneumonia, and Haemophilus influenza to protect us against them, okay? So that is for the glycocalyx. So here we have a checkpoint. Do you guys have any questions related to the external structure of the bacteria? The cell envelope? All good? I know it's a lot of information. It's a very cramped lecture. Okay. So now, since we talked about the cell wall, we're going to talk about the synthesis of the cell wall. Okay. So for the cell wall synthesis, we have two things. We have components. We're going to look at the components of the cell wall, and then we're going to look at the stages of the synthesis of the cell wall. So we already said that peptidoglycan, peptido, so we're going to have proteins, and glycans, so we're going to have sugars. What are are glycans or sugars, since they're the backbone. Usually the last part of the name, this is the main structure, and then the first part is what's added to it. So what are the glycans or our backbone? We have two things, which are NAM and NAG, okay? NAM and NAG are our sugars. This is something acid, murmuric acid, I don't really remember, but this is N-acetyl glucosamine. So it's a glucose molecule that has an acetyl attached to it and an amine molecule attached to it, okay? But you don't really have to remember the thing, you just remember the abbreviations, NAM and NAG. Then we have our peptides or amino acids, protein, which are D-glutamine, L-alanine, D-alanine, and L-lysine or DPA, okay? And this depends, L-lysine usually is in gram-positive and DPA is usually in gram-negative, okay? So D-glutamine, L-alanine, D-alanine, and L-lysine or DPA. So what are the stages? These are the components. So what are the stages? First, we have the cytoplasmic stage. Cytoplasmic stage means that it happens in the cytoplasm. And what do we need to do in the cytoplasm? We need to form our components. We need to form our precursors. So that's what's going to happen in the cytoplasmic stage. And this stage is usually inhibited by two antibiotics, which are fosfomycin and cycloserine. And we're going to look at how those are inhibited. Then we have our second stage. So we moved on from the cytoplasm out to the cell membrane. So the membrane stage. What happens here? Here, we're going to have to transfer the precursors from inside to outside because the cell wall is outside the cell. So we're going to transfer from inside to outside, okay? And then when we transfer it to outside, we need to incorporate it into the already forming peptidoglycan chain, right? Okay, and this step is inhibited by vancomycin and bacitracin. Again, we're going to look at how those inhibit. Lastly, now that we left outside, we're going to have the extracellular stage, which is outside the cell. And here we're going to have to cross-link the chains with each other to form a strong one peptidoglycan chain, okay? Peptidoglycan cell wall. And this stage is inhibited by beta-lactams. Again, we're going to look how did they do that, okay? So starting with the cytoplasmic stage, we said we need to form our precursors, okay? So our first precursor is NAG, okay? And where does NAG come from? We said it's basically a glucose molecule that has an acetyl and amine attached to it, okay? So NAG forms from glucose. Here we have our first precursor. Our second precursor that we have to form is NAM, right? So we have NAG and NAM, our sugars. How do we form NAM? First, NAG through UTP is going to be converted to UDP NAG. UTP becomes UDP and attaches to NAG, and that's the first step. And then now we have to convert this NAG into NAM. How do we do that? We have an enzyme called enol pyruvate transferase. I'm sorry, it's supposed to be transferase. That's going to convert NAG into NAM, okay? So, and this step is the step that's inhibited by fosfomycin, okay? So fosfomycin inhibits this enzyme, and therefore we're not going to be able to have our sugar, which is NAM, so we're not going to be able to form the cell wall, okay? After that, we already have our sugars, now we need to have our peptides. And the peptides, they're going to attach to NAM. So the way I kind of remember it, I'm not sure if it's really good, which sugar does the peptides attach to? I would think that if you sleep a lot and you slept a lot, you gain a lot of weight. I don't know, like, you know, you're not moving. So you're gaining the peptides. So the peptides added in the first step is L-alanine, D-glutamine, and L-lysine. These are the first peptides that are added. There are 3 peptides. So we're going to have a NAM NAM tripeptides, okay? And then now we're going to have to add more amino acids. And these are the D-alanine amino acids. So first we have L-alanine, D-glutamine, and L-lysine. Then we're going to add D-alanine, and we're going to add 2 molecules of D-alanine. We're going to have 5. So now we formed our NAM pentapeptide, UDP NAM pentapeptide, okay? And D-alanine is inhibited by cycloserine. I wouldn't say it's inhibited, but basically what happens is that usually in the environment, the alanine is present as L-alanine. It's not present in D-alanine most of the time. And so the bacteria has to convert L-alanine to D-alanine. How does it do that? It converts it through an enzyme called racemase. And this racemase is inhibited by cycloserine. So if you inhibit cycloserine, you're not going to have your NAM pentapeptide, and therefore you're not going to have your precursors for forming the cell wall. So is the cytoplasmic stage all clear? Move on to the next stage. Okay, so now we have the membrane stage. What happens in the membrane stage? We said we need to transfer the precursors from inside to outside. And how does this happen? We have a lipoprotein that's called bactoprenol. So bactoprenol, the precursors, which is NAM and NAG, NAM pentapeptide and NAG, are going to attach to the bactoprenol, and then they're going to be transferred outside the cell. So basically, bactoprenol is the driver that drives it outside the cell. And this is with the help of an enzyme called peptidoglycan synthase, which is also going to attach the chains to each other, linearly attach the chains to each other. So what happens? Bactoprenol carries NAM and NAG, NAM pentapeptide and NAG outside, and then they're going to be linked. And this is with the help of peptidoglycan synthase. Now, the bacterial cell has a limited supply of bactoprenol. So it needs bactoprenol to go back inside the cell. So how does bactoprenol go back to the cell? So goes back to using the enzyme phosphatase. So phosphatase is going to kind of remove a phosphate molecule from bactoprenol and allows it to go back to the cell, okay? So here, what's inhibited? Basically, first we have vancomycin. And the reason I put vancomycin in the pink D-alanine molecules is that vancomycin inhibits the transfer of NAM and NAG outside the cell, however, not directly, not through inhibiting peptidoglycan synthase or inhibiting bactoprenol. It basically clings onto the D-alanine molecules, and by clinging onto D-alanine, it's going to prevent the transfer. And if it prevents the transfer, then you're not going to be able to form the cell wall synthesis, right? And vancomycin is a very, very important antibiotic. Where can you use vancomycin? What is it good at killing? MRSA, perfectly. So if you have MRSA, you can kill it using vancomycin, okay? It's a very potent gram-positive killer of bacteria, okay? Yeah, antibiotic. Then, so this is transfer outside. What about having the bactoprenol go back inside? We said we have limited supply, so we need the bactoprenol to go inside. And we said it's with the enzyme phosphatase. So what's an inhibitor of phosphatase? Then this enzyme is called bacitracin. Not enzyme, drug is called bacitracin. So bacitracin inhibits phosphatase, and therefore bactoprenol is not going to go inside, and you're not going to have enough supply. Then you're not going to be able to transfer molecules outside, and you're not going to be able to form your cell wall. Okay, and bacitracin is important to differentiate between two types of bacteria. Do you guys know which ones? Two bacteria that you can differentiate with bacitracin. So for example, one would be sensitive to bacitracin and the other one would not be sensitive to bacitracin. And then you're going to be able to know, okay, yeah. Yes, perfect. So Strep pyogenes is bacitracin sensitive and Strep agalactia is bacitracin resistant. So then if you did, for example, blood agar, you found that the bacteria is beta hemolytic, and then you put bacitracin. If the bacteria die, then it's Strep pyogenes. If they don't die, then it's most likely Strep agalactia, right? To differentiate between both of them. Okay. Last stage, which is the cross-linking. So this is the third stage, and it's the extracellular stage, okay? So here, now that we formed the linked chain, the linear chain, we now need to cross them with each other, so cross-linking them. And how does cross-linking happen? It happens through an enzyme called transpeptidase. Another name for it is penicillin binding protein, okay? So transpeptidase helps cross-link the peptidoglycan chains to each other to make them stronger. And this usually happens from the third amino acid of the one of the pentapeptides, and it attaches to the D-alanine, which is the fourth amino acid in the other one, okay? So that's how it happens. So that's basically the transpeptidase stage. And this is inhibited by beta-lactams. So beta-lactams, one of them is penicillin, right? And that's why the other name, which is penicillin binding protein. So they attach to it, inhibit the cross-linking, and therefore they're going to inhibit the cross-linking of cell wall, making it weaker and so on, okay? So one more thing is that there's a difference between the cross-linking of a gram-negative bacteria and a gram-positive bacteria. In gram-negative, it's a thin peptidoglycan wall, so it doesn't need that much cross-linking. So it's pretty much just a direct cross-link between this third amino acid and the D-alanine, okay? But in gram-positive, you're going to need a stronger cross-link. So you're going to have 5 molecules of glycine that are going to cross-link the amino acids between each other, the peptide chains, okay? So that is for the peptidoglycan synthesis. So now I want you guys to tell me the steps, like the first step, the second step. I'll give you the first step, which is NAG forms from glucose. Then what happens? Go. You can unmute yourself if it's too long to type. Does NAG to NAM form immediately? What happens first? Okay, NAG, yes, NAG to UDP NAG. And this is through what, like, what is it? UTP, yes, perfect. Okay, so step 2, NAG converts to UTP NAG, UDP NAG by UTP. Then what happens? What happens after UDP NAG? UDP NAG, okay, to NAM. And this enzyme is? Yes, okay, thank you. I wrote these wrong. I'm going to fix them. But yeah, the transferase, okay? So then after that, what happens? And we said this enzyme is inhibited by, enol phosphate transferase is inhibited by which drug? Phosphomycin. Yes. Is it? Yes. I'm going to check it. Get back to you guys. 1 second. Yes, 1 second. I'm really sorry. Okay, it is enol pyruvate transferase. So here it was right there, I wrote enol pyruvate phosphate. Okay, so it's enol pyruvate transferase. Thank you. So yeah, we said it's inhibited by phosphomycin. And then step 4, I put it by mistake. What happens? We formed our sugars, the NAG and the NAM. Then we have to form, adding the amino acids, and the amino acids are added in 2 steps. First, what do we add? What are like the first 3 amino acids that we add? L-alanine, D-glutamine, L-lysine, or yes, perfect. Okay. And so if we're going to form UDP NAM tripeptides, 3 peptides. And then what are we going to do? What amino acids do we add next? Yes, add the 2 D-alanines, and you're going to form UDP NAM pentapeptides, right? So now we formed our L-lysine and DPA is part of the 3 amino acids that are added first, forming the tripeptide. And then D-alanine, we said first L-alanine is converted to D-alanine through racemase, which is inhibited by cycloserine. And D-alanines, 2 D-alanine molecules are added, forming the penta 5 peptides, right? UDP-NAM pentapeptides. Okay. Then, what happens next? We formed our precursors. What do we have to do next? We finish the cytoplasmic stage. Membrane. So what happens? Transfer it across the membrane using what? Bactoprenol. Okay, perfect. With the help of what enzyme helps bactoprenol transfer? Yes, peptidoglycan synthase. Okay, now what do we do next? Bactoprenol is out. How do we bring it in? What enzyme? Phosphatase. Perfect. Okay, so phosphatase allows the bactoprenol to go back to the cytosol. And then now we have our linear chains. What do we do? What do we need to do next? Last step. Yes. Okay, so we need to cross-link with the help of transpeptidase. And if we're going to just remember the inhibition, so enol pyruvate transferase phosphomycin, the D-alanine racemase inhibitor is cycloserine, inhibitor of transferring outside by clinging onto D-alanine is vancomycin, and then phosphatase inhibition by bacitracin, and then transpeptidase is inhibited by beta-lactam. So all of these enzymes inhibit cell wall synthesis, and therefore they can kill the bacteria by lysing it. Okay? So now we reached that checkpoint. Is everything clear until now? Before we move on. 1 second. This one or this one? This one? Yes, okay. So here we said that the gram-negative bacteria, they have a thinner peptidoglycan, remember. So if they have a thinner peptidoglycan, they don't need that strong of a cross-link, and the cross-linking is just a direct bond between the amino acids of the facing peptidoglycan chains, okay? However, in a gram-positive bacteria, the cell wall is very thick, and it needs a stronger cross-linking. And what happens is that you have 5 molecules of glycine that are going to cross-link the help cross-link the peptidoglycan chains with each other. So this is basically a direct one. This one is a chain of 5 glycine molecules in gram-positive bacteria, which makes sense. It needs a stronger bond because it's a thicker peptidoglycan layer. Does that make sense? Or do you have another question? Oh, yeah. Okay. So basically what happens is that originally there are 2 D-alanine molecules. We said it's a pentapeptide, right? But then what happens is that when the 3rd amino acid binds to the target, I would say the target other amino acid, which is the 4th D-alanine, you lose a D-alanine. So what happens is that you have 5. When this bond happens, a D-alanine is lost. But for this bond to happen, you need the 2 D-alanines. If you don't have 2 D-alanines, this bond cannot happen. But when the binding happens, the D-alanine molecule is lost. So basically, the 5th D-alanine molecule is there to help in this bonding. And then when the bond happens, halas, it's not needed anymore and it leaves. Does that make sense? Okay. No problem. Okay. Is there any other question? How does vancomycin work? Okay. So vancomycin basically we have these two D-alanine molecules, okay? And it's going to bind to the D-alanine molecule. By binding to the D-alanine molecule, what happens is that they're not going to be able to be transferred outside the cell through bactoprenol. So basically, it inhibits the transfer of the precursors from inside the cell to outside the cell. And if you're not able to transfer them outside the cell, then you're not going to be able to have a cell wall outside, and you're not going to be able to do cell wall synthesis. So basically, vancomycin is a cell wall synthesis inhibitor. It's under the class that's called glycopeptides. Binds to the D-alanine, no transfer. It's not outside the cell, you can't form a cell wall. Did that answer your question? No problem. Is there any other question? All clear? You can move on. Okay, so we'll move on to the next part. So we did the internal structure, and then we did the cell envelope, and now we're going to look at the appendages, okay? So starting with the flagella. So what is a flagella? Flagella is a long filamentous appendage. As you can see, it's very long, okay? It is made up of flagellants. A trick to remember what things are made up of, if it's flagella, it's flagellants, and then we're going to see other examples. Just add illins to it, basically, okay? So flagella is made up of flagellants, and it has H antigens. So whenever a bacteria has an H antigen, know that it has a flagella. Okay, so flagella stands for H antigens, or H antigens stand for flagella. And if it has an antigen, then we can form antibodies against this antigen, right? Which is important for us because then we can attack the bacteria, okay? And what's the function of a flagella? It allows for the movement of the cells, okay? So motility is helped with the flagella, okay? And what is the anatomy of a flagella? First, we have a basal body, and this basal body is inside the cytoplasm, and it's basically the rotating disc. This is the part that basically moves, okay? So the basal body moves, and this is needed with the help of ATP. So we need energy, which makes sense. You need energy to move. So the basal body needs ATP to move and rotate, okay? Then we have a hook, and a hook is in the cell wall part of the bacteria, and it's going to connect it to the outside, which is the helical filament, okay? So there's the basal body in a hook that hooks the basal body to the helical filament, which is the part outside the cell that is moving. So the basal body is the one that rotates and causes the thing, and then this motion is transferred to the filament outside, and now you can move, and the bacterial cell can move, okay? And now types of flagella, I wouldn't say types of flagella, rather than the arrangement of the flagella is basically divided into 4 types. First, we have monotrichous. Trichous means hair. So mono, one flagella, one flagella at one pole or one end of the bacteria. Then we have amphitrichous. Amphi means both. Kind of like an amphibian is a frog. It can live in both water and land. So amphi means both. So there is one flagella at both ends of the bacteria. Then we have lophotrichous. Lopho means that there's 2 or more flagella at only one end. I don't know if this is going to help you remember it, or this is more confusing, but lopho is basically some animals have these like spikes or hairs on top of their head. So that's what lopho is. So you can think of it as like the bacteria having hair from only one end, lophotrichous. And then we have peritrichous. So peri is all around, periphery. So this has flagella all around the cell, okay? And then there's endoflagella. Endo means inside. So this time the flagella, instead of being outside the cell, it's within the cell, within the outer membrane. And we're going to talk more about these endoflagella, okay? And another thing that's very important is that if a bacteria is very motile, so it moves a lot, it can show you something that's called swarming when you culture it on an agar. So this is swarming, basically, as you can see, okay? This is swarming of a bacteria. Do you guys know an example of a bacteria that shows swarming on agar? Oh, mashallah, that was very fast. Yes, Proteus, okay? So Proteus is one of the bacteria that shows swarming. So that means it's very, very motile, right? Okay. So what would we call this bacteria, this arrangement of flagella? Monotrichous 1, okay? The second one, Lophotrichus, perfect. So there's 2 or more from only one end, just like the hairs of the animals. It's Lopho. This one, yes, okay. This is Amphitrichus and this is Peritrichus. So Amphi, as you can see, there is 1 at both ends, okay? And I made it green to remind you of a frog. I don't know if that's gonna help. But then there's Peri, which is all around the cell, okay? So basically these are the arrangements. Then here we have endoflagella, and we're going to talk more about the endoflagella. So endoflagella, we said that the flagella is within the outer membrane of the cell. If it's within the outer membrane, is endoflagella present in gram-positive or gram-negative bacteria? Yes, so endoflagella is only present in gram-negative bacteria. You don't find endoflagellum in gram-positive bacteria because it's within the outer membrane, okay? And we had helical filament before, that's outside. This one forms an axial filament. So endoflagella is an axial filament, okay? And it moves through torsion, basically, okay? So that's how it moves. And it's present in cells that look like this, which are called spirochetes, spirosheets, whatever you want to call it, but they're spiral and they have endoflagella within them and they spiral around in torsion, okay? So what are some examples of spirosheets? What does axial? It's basically just the flagella, but it's within the outer membrane. Like it's not outside the cell, it's inside the cell. I don't know if that makes sense. Yeah. Okay. So what are some examples of spirosheets? Treponema. What's the disease of treponema? What disease does treponema cause? Syphilis, yes, okay. And what's the other bacteria? Another one? Okay, Leptospira is also one. And another one? Last one. Okay, so Borrelia burgdorferi, I don't know how to pronounce this one, but Borrelia, which is Lyme disease, the one that's transferred through ticks and has the bullseye rash. And then there's the Treponema pallidum, which is syphilis, and as you said, Leptospira too, okay? And they look like this. They're a spirosheet, okay? Now let's look at fimbriae, okay? So for example, these are fimbriae, and they're hair-like structures, short and thin hair-like structures, appendages, and they're present in both gram-positive and gram-negative bacteria, and they're made up of fibrillins. Remember, flagella is made up of flagellins. Fimbriae are made up of fibrillins, okay? You just added the lins in the end, okay? And what codes for fimbriae are chromosomes, okay? So fimbriae are found in both gram-positive, gram-negative, made up of fibrillins, and they're coded by the chromosomes. And what is their function? They help in adherence. So therefore, it's a virulence factor. If you have fimbriae, you're able to adhere to membranes and tissues and basically colonize them better and cause infections. And therefore, it's a virulence factor. So fimbriae helps in adherence, okay? Another thing very important about fimbriae is that they undergo antigenic variation. What does antigenic variation mean? What do you guys think antigenic variation means? Exactly. So they're able to change kind of their antigen. So for example, they had an antigen. Your body recognized this antigen, formed antibodies against this antigen. But then now they underwent antigenic variation, changed these antigens. So the antibodies that you had are no longer effective. And now you have to form new antibodies against these new antigens. So it basically helps it in escaping the immune system. So fimbriae has two things, two virulence factors, adherence, and another thing is the antigenic variation, okay? So now there is pili. So what are pili? Pili are also thin hair-like structures, okay? However, they are longer and thicker than the fimbriae, okay? So they're shorter than a flagella, they're thinner than a flagella, but they're longer and thicker than a fimbriae, okay? And they're mainly present in gram-negative bacteria. So we said that fimbriae are mainly in gram-positive and gram-negative. They're in both, but pili are mainly in gram-negative bacteria, okay? And they're composed of pilin, flagella, flagellins, fimbriae, fimbrillins, and pili, pillins, okay? And the origin or what's coded, what codes for it is a plasmid, PP, okay? So plasmids code for pili, chromosomes code for fimbriae, okay? And why are pili important? We have two types. First one is the sex pili. So this one is basically, it helps the conjugation, which I said, if you remember, we said that conjugation, plasmids transfer through the process of conjugation. So this is with the help of pili, which makes sense. So plasmids code for pili that help them to transfer, right? So this is conjugation. What happens basically is that a bacteria that's F positive, we said fertility positive, so it has a plasmid and it has the pilus. And then there's a bacteria that has no plasmids and no pili, so it's F negative. They're going to, this bacteria is going to replicate its plasmid, and then it's going to attach and it's going to basically with the pili form like a tunnel. So basically the pili acts like a tunnel or a bridge, okay, among which the plasmid is going to transfer to the other bacteria. And now this bacteria that had no plasmid now has a plasmid, okay? And why is this important? For example, this bacteria has a plasmid that codes for an antibiotic resistant gene, and this bacteria doesn't. So this is resistant and this is not. Now, this bacteria can transfer its resistance to another bacteria. So this is good for the bacteria, but it's bad for us, okay? So basically, these are done by the sex pili. Then we have the common pili. So the common pili are basically another name for the fimbriae. So some people refer to fimbriae and pili as the same thing. However, for the sake of your exam, remember fimbriae as fimbriae and pili as pili, okay? But common pili are basically fimbriae, so the same function, which is adherence mainly, okay? So now let's revise the changes. So fimbriae, what's the structure? They're short and thin, and pili are longer and thicker. Okay. What about the number? I did not mention this, but maybe you can tell from the images. Fimbriae are more, okay, yes, and pili are fewer in number. Okay. What about the origin or what encodes for it? chromosome for fimbriae, and pili? Plasmids, okay. As I said, PP, okay. And then composed of, like, what's the proteins? Fibrillin and pili? Pillins and flagella? Flagellants, okay. And fimbriae are more in which type of bacteria? Gram-positive or gram-negative, or both? Both. And pili? In gram-negative. Perfect. And what's the virulence factor of fimbriae? Give me two. Here I wrote one, but two. Adherence and antigenic variation. Perfect. And pili? Oops, sorry. Oop. I mean, you saw it, but basically it helps in antibiotic resistance because you're able to transfer it just through conjugation. Okay. So this is for the appendages. Now we reach the second point. Do you guys have any questions related to anything that we took until now? All clear. Perfect. Okay. So I think Aisha, it's Aisha then right now. So do you guys want to take a break or do you want to continue? Yes. Okay, no. It's not the fact that it's antigenic that's a virulence factor for the fimbriae. The thing that's making it a virulence factor, it's that it's the antigenic variation, which basically means that it can change its antigen. So basically, for example, you have this fimbriae or this bacteria was there. It had the antigens on the fimbriae. The immune system recognized these antigens, produced antibodies against these antigens, and now you should be able to kill the cell, right? But then what happens is that it undergoes antigenic variation and it changes its antigens. And now the antibodies that you already have for this bacteria can no longer work on it, and you need to form new antibodies. So it basically escapes the immune system and it escapes the antibodies that are forming against your against this bacteria. So this is the virulence factor, the antigenic variation. As for antigens itself, if something being antigenic, I would not say that that's a virulence factor. Instead, that's something helpful to us since we can recognize them and kill them. What's remaining? So we have atypical bacteria, and then staining techniques, and then we have endotoxins, exotoxins, and then endospore formations, and then some questions. 4 things. Yes. Okay. So you guys want to like take a break and then continue? Or do you want to finish in one go? So they continue? Okay. It's up to you guys, but okay. We can continue. Okay. So now let's move on to atypical bacteria. So atypical bacteria means that we cannot classify it mainly into a gram-positive or gram-negative. So the main factor is that we can't really stain them, and there's something different about them. They don't fall into the category of gram-negative or gram-positive most of the time. So first thing we have is mycoplasma, okay? So mycoplasma, what's atypical about mycoplasma? Number one, it has no cell wall, which is very weird. All bacteria have cell wall, but mycoplasma has no cell wall. And instead of a cell wall, it has sterols in its cell membrane. So we know that the cell membrane of a bacteria lacks sterol except mycoplasma. It has sterols, so no cell wall and sterols, okay? And since it lacks a cell wall, then the antibiotics that work against cell wall synthesis or work against the cell wall and inhibit cell wall formation do not work on mycoplasma because mycoplasma does not even have a cell wall. So basically, mycoplasma has an innate and inherent resistance to cell wall synthesis inhibitor, which is good for mycoplasma. So only certain types of antibiotics work against mycoplasma. Can any of you guys give me example or a class of drugs that works against mycoplasma or atypical bacteria in general? Macrolides, yes, perfect. So there's a mnemonic that's called FAT, F-A-T. So I think basically fluoroquinolones and azithromycin, which is a macrolide, and T-tetracyclines. So all of these work against atypical bacteria. So what are the virulence factors of mycoplasma? It has a molecule called P1 adhesin. So adhesin, it adheres to the respiratory epithelium. Why respiratory epithelium? If you know the full name of the bacteria, for example, Mycoplasma pneumonia, so it causes pneumonia, so it's going to adhere to the respiratory epithelium. P1 adhesin. The other virulence factor is that it has toxic metabolites, and these toxic metabolites are going to cause the damage to the tissue of the host or our tissues, okay? So this is how mycoplasma works. It adheres, and then it releases toxic metabolites. Then we have chlamydia. Chlamydia, what's atypical about chlamydia is that number one, it's technically a gram-negative bacteria. Why? Because it has a cell wall, and above the cell wall, it has an outer membrane. So it kind of resembles in structure a gram-negative bacteria, okay? However, it's an obligate intracellular, meaning that it cannot live outside the host cell. It has to live inside, usually white blood cells. So it has to live inside a cell. And that's why chlamydia used to be thought of as a virus. People used to think that chlamydia was a virus until then they discovered that it was a bacteria because it couldn't live outside a host cell. It was difficult to culture because it needs a host cell, okay? So chlamydia is an obligate intracellular, okay? The other atypical feature is that we said that our cell wall is mainly made up of peptidoglycans. Chlamydia has a cysteine-rich cell wall. So C for C, cysteine, chlamydia. So that's different than the cell walls in other types of bacteria, okay? And as you can see here, we have a picture. These intracellular inclusions, these orange things, are the chlamydia inside cells, okay? So what are the virulence factors of chlamydia? Number one, antigenic variation. So again, we said it can change its antigens. And what other thing has antigenic variation? We mentioned already. Structure, yes, Fimbriae, antigenic variation. And it also has LPS. Yeah, no, it's fine. It also has LPS, okay? So chlamydia has it exists in two states, an elementary state and a reticulate state. So in an elementary body, this is the infective form of chlamydia, and reticulate body is the vegetative state of the bacteria, which is basically, it's just replicating and living and metabolizing and replicating, but it's not infecting. So how do you kind of remember this? I don't know, but if you think about elementary age school kids, their positivity is infective, although they're mostly annoying, but basically elementary, they're infective. Reticulate, they're not infective, they're vegetative, just dividing, okay? Another question, what other bacteria is an obligate intracellular other than chlamydia? You took two, I think. Gonorrhea is an intracellular. but something that's like obligate intracellular. Yes, rickettsia. So rickettsia is another obligate intracellular. The types or the states, basically, the chlamydia exists in two states. First thing is elementary. So when chlamydia is in an elementary body, it can infect other cells. It basically causes an infection. But when it's in a reticulate body, it's not going to infect other cells. It's going to stay where it is, but it's just going to multiply. It's a vegetative state, multiply and metabolize, but there's no infection. It's only going to infect when it's an elementary body. So it kind of has two states. A state that infects and a state that just multiplies and divides. Does that make it clear? Okay, no problem. Okay, so this is for chlamydia. The last thing that we have is mycobacteria. There are these bacilli. So what's the atypical feature about mycobacteria is that they have outside a thick waxy layer made up of lipids. It's called mycolic acid. So mycolic. Okay, so mycobacteria has mycolic acid. And why is this mycolic acid important for the mycobacteria? It allows us to survive harsh environments like extreme pH, dryness, and other stuff. So basically, this mycolic layer allows this mycobacteria to survive, okay? And another thing, this mycobacteria can actually live inside the macrophage. It thrives inside the macrophage, okay? So basically, you know how a macrophage will engulf the bacteria? Instead of being able to kill it, it's not going to be able to kill it. What happens is that the mycobacteria is going to replicate and multiply inside the macrophages, okay? And this can somehow also, it's going to be able to evade the immune system because it's inside the cell. It's not outside the cell, okay? And it doesn't really stain with the gram stain because of this thick waxy layer, okay? Instead of gram stain, we're going to stain it with something called acid-fast or Ziehl-Neelsen stain, okay? And if it stains with acid-fast stain, then you immediately know that this is a mycobacteria rather than other types, okay? So here, another match game, like matching. So which bacteria is an obligate intracellular? Chlamydia, okay? Which bacteria has P1 adhesion as a virulence factor? Mycoplasma, okay? Which bacteria has an elementary and a reticulate body? Chlamydia. And which one is the infective and which one is the vegetative? Elementary is infective, yeah. Yes, okay. So the reticulate is going to be the vegetative, okay. Oops, sorry, which one lives in macrophages? I mean, you saw that. Mycobacterium. Which one produces toxic metabolites as a virulence factor? Mycoplasma, perfect. Which one is technically a gram-negative bacterium? Chlamydia, perfect. Okay, which one has a thick lipid waxy layer that's called mycolic acid? Mycobacterium, and it allows us to survive against harsh environments, okay? Which one can undergo antigenic variation? It's able to hide inside the cells. Which one? You have to be sure. Yes, okay. Chlamydia can undergo antigenic variation. Which one has no cell wall? Mycoplasma, perfect. Which one can survive in harsh environments? Mycobacterium. Perfect. Okay. Which one has sterols? Usually we lack sterols, but which one has sterols? Mycoplasma, yeah. No cell wall, but has sterols. Which one has cysteine-rich cell wall? Chlamydia. So CC, cysteine. Okay, which one can stain with acid fast or Thiel-Nielsen stain? Mycobacterium. Perfect. Okay. And the last thing, which one has an inherent resistance or innate resistance to cell wall synthesis inhibitors? Mycoplasma, because it doesn't have a cell wall. Okay, perfect. So checkpoint. Do you guys have any questions regarding the previous? Are all clear? P1 adhesin, yes, for mycoplasma, it adheres to the respiratory epithelium. That's why mycoplasma pneumonia can cause pneumonia. Okay, no problem. Any other question? Oh, good. Do we move on? You want to take a break? Sure. Yes, okay. We can have a 10, 15-minute break, kind of. Okay? Okay, we'll be back later. Okay, so at what time? So around like 7:10, okay? Be back. Is that enough for 7:15 or 10? Okay. Around 7:10. Tamam. Okay. Thank you guys. Okay. So now let's move on to the next concept, which is bacterial toxins. No, I think I went too far. Staining techniques. Okay. So let's look at the staining techniques. So we know about the gram stain and the acid-fast stain. So let's look at how they're done. Starting with gram stain, it's very simple. First, we have something that's crystal violet. From its name, violet, so it's purple. So you add this to the slide that has the bacteria. You add the crystal violet stain. All bacteria, whether they're gram positive or gram negative, are going to stain purple with the crystal violet, okay? But then what do? You're going to put iodine. When you put iodine, again for 1 minute, so 1 minute, and then you rinse off, and then 1 minute, and then you rinse off, what iodine does is that it's going to fixate the crystal violet to the cell wall. So if something has a cell wall, a very thick layer of cell wall, for example, gram positive, it's going to be more fixated than something like gram negative, which has a very thin cell wall, okay? So first thing, crystal violet stains everything. Iodine fixates it into the cell wall by making the crystal violet less soluble, okay? And then after that, you're going to add alcohol. Alcohol is a decolorizing agent. So it's going to remove the color, except what's fixated. So which one has the thicker cell wall that's going to fixate the dye more? Gram positive or gram negative? Gram positive. So this means that after putting the alcohol, which is a decolorizer, the stain is going to wash away from gram negative bacteria, but it's going to stay in gram positive bacteria, okay? And after that, we need to see both colors, so we're going to add a counter stain, and the counter stain in this case is safranine. Safranine, just like saffron, is red, so you're going to add the safranine, and whatever wasn't stained by crystal violet, which is gram negative, is now going to stain with safranine, and it's going to be red or pink. It's the more accurate one is pink. So gram negative will end up being pink, and gram positive will end up being purple, okay? So these are the steps of gram staining. First crystal violet, then for a minute, then iodine for another minute, and then alcohol for 10 seconds, and then safranine for another minute. So Is this gram-positive or gram-negative? Positive. Bonus question. Is this a staph or a strep? Yes, staph, because it's in clusters. Strep would be in chains. Okay. What is this one? Gram-positive or gram-negative? This one is gram-negative because it's pink. Okay, so gram-positive and gram-negative. That's basically it. As for acid-fast or Ziehl-Neelsen stain, this one we said to stain which bacteria? Which bacteria stains acid? Yes, Mycobacterium. So basically another name for Mycobacterium is it's an acid-fast bacilli because it's a bacilli, it's rod-shaped, and it stains with acid-fast stain or Ziehl-Neelsen stain. Okay, so here first you're going to add something called carbonyl fuchsin. It's fuchsia, it's pink in color. Okay, so you're going to add this carbonyl fuchsin. When you add it, everything in the sample is going to stain pink. Okay, and then now you're going to apply heat. When you apply heat, this is going to fixate the dye in the sample, in the bacteria. Okay, so first you're going to add carbonyl fuchsin to stain, and then you apply heat to fixate it. In gram stain, we did iodine. Here we're doing heat. Okay, and then we're going to add alcohol again as a decolorizing thing. Okay, so what happens basically is that in the Mycobacterium, the mycolic acid layer basically kind of covers the stain. So now the stain is fixated. So even when you add alcohol, it's not gonna wash away. So the acid-fast bacilli is going to stay pink, but whatever else is there is going to end up decolorizing. And now you need to add a counter stain to see the other things. So what are you gonna add? Methylene blue. Blue is blue in color. So whatever is not a Mycobacterium is going to look blue. So usually it's the epithelium tissue that's with the sample or with the slide is going to end up being blue, and the Mycobacterium is going to be the pink bacilli. Okay, so basically carbonyl fuchsin, which is fuchsia, it's pink in color, and then you heat to fixate, again alcohol to decolorize, and then methylene blue, which is blue in color as a counter stain. Okay, so for example here, this is the acid-fast bacilli, and all these are the epithelial tissue or whatever that's not a Mycobacterium. Okay, so now organize the steps into what should be step 1, step 2, step 3, and step 4 for gram stain. So which one is the first step for gram staining? Step 3, okay. Crystal violet first. Everything's going to be purple, and then okay, 3, 4, 1, 2. Okay, crystal violet first, and then iodine to fix it, and then alcohol to decolorize it, and then safranine that's going to stain the gram negative. Okay, perfect. So these are the steps in order. Okay, now for acid-fast bacilli. Again, you can put it in order all together. What's first step, second, third, and fourth step? Two, 4, 1, 3. Perfect. Yes. So first carbonyl fuchsin, pink, and then heat to fixate it. It's only going to be in the Mycobacterium layer. And then the alcohol, it's not going to be able to wash the stain away from the Mycobacterium, but it's going to decolorize the others. And then methylene blue, which is going to counter stain the other epithelial tissues and stuff. Okay, perfect. So that's the order, correct order. So now checkpoint. Do you have any questions related to these staining techniques? Clear? Okay, good. Sometimes they ask about this in the OSPE, and sometimes they might ask about it in the MCQs because it is part of the lectures, okay? Now we're moving on to bacterial toxins, okay? We're going to talk about exotoxins and endotoxins. So what is an exotoxin? An exotoxin is basically the toxins produced by the bacteria. The bacteria is still living. It's produced inside the bacteria, but it's released outside. So it's exo, it's released outside the bacteria. and it's going to cause damage, okay? But then we have endotoxins. So endotoxins, they are an integral part of the bacteria. They are basically a structure of the bacteria that are only released when the bacteria die. So exotoxin, bacteria is living, releases the toxin outside, but endotoxin, it's within the bacteria. It's part of the bacteria that's released when the bacteria die, okay? So let's first look at exotoxins and how they work, okay? So exotoxins, mainly, they're made up of proteins, okay? And they're usually named after a bacteria, the name of the bacteria or the site of action. So for example, Shigella has Shiga toxin. That's its exotoxin, okay? Or the site of action, for example, Staph aureus has an enterotoxin. So an enterotoxin works on the enterocyte, so the intestines, right? So Dr. Hana mentioned that if she gives you a question that says which of the following is an exotoxin, look for a bacterial name or a site of action, okay? So that would most probably be the exotoxin, okay? They are made from plasmids, or they're encoded mostly by plasmids, okay? And they're all heat soluble. What does heat soluble mean? What does heat soluble mean? Okay. Yes, exactly. So they can't withstand heat. So if you apply heat, they will die, except two toxins. Staph aureus is enterotoxin, right? So for example, a question comes, there is a person who ate street food from somewhere. The street food was hot, obviously. And then within 3 hours, they started vomiting and so on. Then what would your suspicion be? It might be a Staph aureus enterotoxin, right? So basically, I'm not sure if you took this, but for food poisoning, you can divide it into it being caused by a toxin or a bacteria. If it's caused by a toxin, you would get the diarrhea and vomiting within 6 hours. And if it's after 6 hours or like 24 hours later, that's when the diarrhea and vomiting started. That means that the person ingested the bacteria that then took time to produce the toxins and cause the vomiting and diarrhea and symptoms of food poisoning. So in a question, a person ate street food and he got the symptoms within a short time, like 3 hours, then you would think, oh, Staph aureus produces enterotoxins and it caused the food poisoning, for example, and it's heat stable, okay? Another one is E. coli. One of the E. coli strains, it has a heat stable toxin, but it's also an enterotoxin. So they're all heat soluble except these two, okay? So exotoxins, they have 3 mechanisms of action. So the first type of exotoxins, they act on the cell surface. So they bind to receptors in the cell surface or in the cell membrane, they bind to receptors. And then by binding to these receptors, they're going to cause intracellular signaling pathways and basically cause the damage that way. An example of this, and it's very important to remember the examples, is the superantigen of Staph aureus. So the superantigen of Staph aureus works by binding to receptors on the cell surface, okay? So how does it do that? The superantigen specifically, it's a very bad antigen. Basically, it binds to the T cell receptors and the MHC2 without the need for the antigen presenting cells to present an antigen. So usually what happens, you have a bacteria that came, the macrophage, say, engulfed it, and then it breaks it down and presents the antigen for the T cells. The T cells recognize this antigen, and then they're going to have the T cells replicate specific to this antigen, right? But what happens in a super antigen is that it can bind to this MHC2 outside of the antigen presenting part and the T cell receptor, okay? So then what happens? You're going to have proliferation of the T cells, release of cytokines, interleukin-2, for example, that's not specific to the bacteria, but it's also causing a lot of immune response. And this immune response is causing excessive inflammation, and this inflammation can cause this toxic shock, septic shock, and death, okay? So it's very severe. It basically causes like a very, very, very severe immune response, okay? So for example, here, if you can see, we have an antigen presenting cell, okay? And a naive T cell. usually what happens, the antigen presenting cell presents the antigen, and the T cell receptor recognizes this antigen, and then it starts replicating, okay? And it divides specifically towards this antigen. However, a super antigen can bind outside this antigen specific place, and it's going to bind to the MHC2, these are MHC2 and T cell receptor, and then it's going to cause proliferation. It doesn't need the cells to recognize what the antigen is. It just causes inflammation, okay? So that's how a super antigen works, okay? Then we have the other mechanism of action, which is damaging the cell membrane. So if it damages the cell membrane through two methods, either pores, okay, or lytic enzymes. So if it has pores, then that's going to cause the cells, there's going to be like an imbalance and the cells can lyse, they're gonna break. Or a lytic enzyme, for example, pneumolysin, that's released by strep pneumo. It's also going to lyse the cell membrane, break down the cell membrane, and therefore the cell will lyse, okay? So again, I said it's important to remember in the examples. So pores, for example, Salmonella and Shigella, they can produce pores. And pneumolysin by strep pneumo is a lytic enzyme that works by damaging the cell membrane. The third type is what acts intracellularly. So first, outside the cell by receptors. Second is on the cell membrane. Third, intracellularly, okay? And intracellularly, you need two things, two steps. First, you need to enter the cell, and then you need to damage the cell, okay? And as you can see, it's bipartite. It has two parts. So it has two subunits, an A subunit and a B subunit. So what happens? The B subunit, okay, helps bind to the cell membrane. So B binds to the cell membrane and allows this cell to endocytose and take in the toxin, okay? And then it disassociates and A becomes on its own, and A is the thing that's going to damage the cell, okay? So A is going to go to whatever target it has, and it's going to damage the cell, and therefore the cell is going to be damaged and it's gonna die. An example of bacteria that have this bipartite subunit toxin are Vibrio cholerae and C. diff. So let's look at how a C. diff toxin can work. So first we have the diphtheria toxin, which we said is made up of two subunits, right? A and B. So the diphtheria thingy is going to bind through the B positive, binds to receptor, BB binds to the receptor, okay? Then it's gonna be engulfed and endocytosis, right? What happens with the diphtheria toxin specifically is that after it dissociates into B and A, the acidic conditions inside are going to trigger the A subunit to go into the cytosol, okay? So that's something specific, is that the acidity or low pH is going to make the A subunit go to the cytosol, okay? And now the A subunit can do what it's supposed to do. So in C. diff specifically, it basically inhibits something called elongation factor 2. So elongation factor 2 helps in the protein synthesis. So you inhibit protein synthesis, you cause cell death. So basically, by the end of it, diphtheria toxin, it works by inhibiting protein synthesis through inhibiting elongation factor 2. And that's how it works. This is an example of a bipartite, yeah, okay? So this is for exotoxins. As for endotoxins, they're made up of lipids. Exotoxins made up of proteins, endotoxins are made up of lipids, okay? And they're made from chromosomes. Exotoxins from plasmid, endotoxins from chromosomes, okay? Exotoxins were heat liable, endotoxins are actually heat stable, okay? And we already mentioned that they're integral parts of the cell itself. But specifically, they are it's the lipid A basically it's the lipid A here portion of the lipopolysaccharide in a gram-negative bacteria. So that means that endotoxins are only formed by gram-negative bacteria. So exotoxins, anything, it's fine, but endotoxins, they're lipid A. And lipid A is only found in the outer membrane of gram-negative bacteria, okay? So what happens basically? A bacteria, it lyses, this bacteria dies, it explodes, either due to antibiotics or macrophages that ate it, so the immune response, okay? Lipid A is released, and lipid A is the endotoxin. It's going to activate macrophages and complements. Then there's going to be a release of cytokines. These cytokines are going to cause inflammation, fever, septic shock, and septic shock can cause multi-organ failure, and then the person can die. So it's a very tricky situation. Sometimes you give antibiotics and you kill all the cells, and then the lipid A is released and you get a septic shock, okay? So yeah, basically lipid A is very dangerous. And the cytokines usually that cause fever, like interleukin-1, TNF-alpha, and interleukin-6, I think, but interleukin-1, TNF-alpha. Okay, so basically that's how lipid A works, and it's the endotoxin, okay? Releasing of cytokines, activating complements, and so on, and macrophages, okay? So now let's look at this table that differentiates between exotoxins and endotoxins. So exotoxins, they're composed of proteins or lipids? Protein. And endotoxins? Endotoxins are lipids, yes, which makes sense because it's lipid A. Lipid A is a lipid, okay? Exotoxin is in gram-positive or gram-negative bacteria or both? Both. And endotoxins? Only gram-negative, okay? Because it's lipid A is only present in gram-negative bacteria. Okay, what about the origin? Is it from chromosome or plasmid, exotoxins? Exotoxins are plasmids, and endotoxins? Chromosome, okay. How do we name exotoxins? Usually the naming. Name of bacteria or? Yeah, site of action. So site of action or the name of bacteria, Shiga toxin, enterotoxin, and so on, okay? As for endotoxins, there is no name. It is lipid A, okay? What about exotoxin? Is it heat liable or stable? What are the two heat stable exotoxins? Staph aureus, enterotoxin, E. coli, the heat stable toxin of E. coli. Okay, and endotoxins are heat stable. Okay, so now exotoxin, we said first mechanism is acting on cell surface. Type 1 exotoxin? Do you mean the one acting on cell surface? Sure. Sorry. Ooh. Okay, sorry. Okay, so as you can see, the super antigen, basically what happens normally is that an antigen presenting cell is going to engulf the bacteria, it's going to break it down, and then it's going to present the antigen of the bacteria to a naive T cell, to a naive T helper cell, okay? Then this naive T cell is going to, through the T cell receptor, it's going to bind to this antigen that's presented by the antigen presenting cell. And what's going to happen is that it's going to form a response, and it's going to basically form T cells that are specific to this antigen that was presented by the APC, right? So it's kind of a controlled reaction just against this antigen presenting cell, and against this antigen, okay, of this bacteria. However, in a super antigen, it does not need to be engulfed by an APC presented as an antigen. No, it basically binds to the MHC2 and to the T cell receptor in a region that's not the antigen variability region. No, it attaches outside, and it causes the bacteria, it causes the T cells to divide. Okay, it causes an immune response that's not specific to any antigen. Okay, it's just an immune response that's going to release a lot of cytokines, activate the immune response, activate the immune system very excessively, and that's going. A lot of inflammation is bad for the body. Okay, it can cause septic shock and so on. Okay, and that can cause death. So that's how a super antigen works. Plus, super antigens, although it's not mentioned here, but it's one of the methods through which autoimmunity can form. Autoimmune, which means the immune system attacks the body, right? So super antigen, because the T cells that are divided are not specific to anything, they can also attack the body. Okay. So yeah, no problem. Okay, so let's go here. Okay, so the first mechanism we said acts on cell surface. An example of that is, we just mentioned, Staph aureus super antigen. Perfect. Okay, then the second mechanism of action of exotoxins, how do they work? First outside the cell, then in the destroy the cell membrane. Okay, so what are examples and how they work? Strep pneumonia, how does it work? What's the toxin and how does it work? Pneumolysine, what does pneumolysine do? Pneumolysine is not pores. No. Pneumolysine just breaks down the membrane. Yes. But pores, which bacteria produces pores in the cell membrane causing lysis again? Okay, so basically pores, Salmonella, Shigella. Okay, so Salmonella and Shigella can produce spores. And lytic enzymes are basically pneumolysine by pneumonia, as you guys mentioned. Okay, so pores, Salmonella, Shigella, super antigen, Staph aureus, and lytic enzymes. Okay, last thing is bipartite. Someone mentioned the B binds and A damage. Yeah, you can think of it as B binds and A attacks. Okay, so what's an example of bipartite? Two examples, two bacteria, diphtheria and cholera. Yes, and what triggers the A subunit to go to the cytosol or travels towards the center of the cell or goes to the cell? High pH, low pH, yes. Acidity, I think probably, yeah, but the more acidic it is, the less pH. The lower pH, the more acidic, yeah. Okay, so Corynebacterium diphtheria and Vibrio cholerae. Remember that B binds and then endocytose, and then A is the attacking subunit. Okay, so now we reach the checkpoint. Do you guys have any questions? Or all clear? All clear? Any questions? Okay. So now, also we're almost done. I know it was a very long lecture and it's very crammed, but yani, inshallah, you guys can revise later. Okay, so last thing is endospores. Okay, so as you can see, endospores are basically metabolically dormant variants of the bacteria, but they're still viable. So it's the bacteria, but it's dormant. It doesn't need to metabolize, but it's still viable, which means it's not dead. It's still living and can still cause an infection later on, which is very dangerous for us. So if a bacteria is able to form spores, that means that it can, when it finds that the environment is not suitable, it's going to form a spore. And then when the environment becomes suitable again, it's going to return back to its original infective state and causes problems. So instead of dying, it forms a spore, okay? So they're not formed by all bacteria, so it's kind of like a virulence factor. And it forms when there are harsh conditions to the bacteria, for example, extreme temperatures and stuff like that, okay? And then when the environment becomes favorable, when the conditions are favorable, it's going to regerminate, okay? So that makes sense? So this endospore can protect the bacteria against extreme temperatures, organic solvents, and boiling, okay? Extreme temperatures like extreme hot, extreme cold, organic solvents, and so on. However, endospores cannot survive something called autoclaving, okay? So what happens in autoclaving? What do you do? Basically, you're going to heat the sample for 121 degrees Celsius for 15 to 20 minutes under steam pressure. So 121 degrees Celsius for 15 to 20 minutes under steam pressure. That's autoclaving. And this can kill an endospore. Otherwise, these things, they're not going to kill an endospore. It's going to be living. It finds a good condition. It can regerminate, okay, instead of dying. And the resistance of an endospore is linked to it having calciums and dipicolinic acid, okay? So these might be related to why an endospore is very resistant, okay? An example of bacteria that have endospores are, for example, Clostridium, which is an obligate anaerobe. So an obligate anaerobe that forms spores, that's Clostridium. An obligate aerobe that forms spores is anthrax, okay? So this is for endospores. So how do they form? So first, what happens is that this is the normal, it's dividing and whatever, but then the conditions are no longer favorable, okay? Doesn't want to die, so it's kind of like hibernating, just like bears, until it finds a good condition. So that they're going to replicate the DNA. After it replicates the DNA, the mother DNA stays somewhere, and the daughter DNA goes to one pole of the cell, and it's called a forespore, and a septum forms, okay? And basically, these cytosol or something, it basically engulfs this forespore, okay? After it engulfs the forespore, you're going to have a peptidoglycan layer forming, and then you're going to have calcium going in, which we said calcium is one of the factors that makes it tough and might explain the resistance of an endospore, okay? After the calcium goes in, the cell can degenerate, and also the DNA degenerates and everything degenerates, and we end up having only the spore. And now the spore is able to resist everything except an autoclave until it meets good conditions. Then it's going to regerminate and it's going to become viable and go into this cycle, and it infects and so on, okay? So basically, first there's division, a forespore forms, then it's engulfed, and then it has calcium and peptidoglycan going in, making it tough. And then the original cell degenerates and the free spore is released, okay? So that's basically how an endospore forms. Also something that's kind of interesting, but basically sometimes you're able to identify bacteria by seeing the position of the spore. So is the endospore in the middle of the cell, or is it in the terminal ends of the cell, and so on, okay? So checkpoint, is endospore clear for now? And we can move on to the questions. Okay, So now we're going to have some questions. You guys answer. You can unmute yourself if you want to read the question out loud. Out loud. Yeah. Okay. So a group of teenagers became ill with nausea, vomiting, severe abdominal cramps, and diarrhea after eating undercooked hamburgers from a local restaurant. 2 of the teenagers were hospitalized with hemolytic uremic syndrome. E. coli 0157 H7 was isolated from the patient's stool as well as from uncooked hamburgers. The H7, H antigen, refers to which bacterial structure? As you can see, you can answer this question without even reading the full scenario, just the last line. Okay? Yes, so flagella. Lipopolysaccharide is a PS capsule, fimbriae. Okay. Yes, so flagella. Correct. Okay. The initial step in the process of many bacterial infections is adherence to the organism to mucous membranes. The bacterial component that mediates adherence is pili. Remember how we said that pili have 2 types, the sex pili and the common pili. So this would be the common pili, which is, what's the other name for the common pili? Fimbriae. Yeah. So if it comes in an exam, just remember that sometimes they will call fimbriae a common pili. Okay, so we said lipid A is the endotoxin that causes inflammation, right? Nucleoid is just a chromosome. Peptidoglycan is the cell wall, has nothing to do with adherence. And plasmid is important for antibiotic resistance, not adherence. Okay. The next question, which of the following is a key structural feature that distinguishes gram-negative bacteria from gram-positive bacteria? C. Why not A? Yes, true. So in an exam, sometimes questions like this can confuse you because you might put the feature of gram-positive instead of the gram-negative. So gram-negative has the outer membrane. The thicker peptidoglycan is in a gram-positive bacteria. Cytoplasm is present in every single cell. And the lack of cell wall is not in gram-negative bacteria. It's in mycoplasma. Okay. A surgery patient at risk for gram-negative sepsis is part of a research project to prevent endotoxic shock using monoclonal antibodies to inhibit the endotoxin molecule. What is the active component of the endotoxin molecule against which the antibodies? Yes, lipid A. Perfect. Copolysaccharide was just the negative component. N-acetylmuramic acid, this is the name I was looking for. That's NAM. That's just a precursor for the peptidoglycans. Oocyte chain, we said, is the antigenic. And teichoic acid is in gram-positive bacteria. Okay? Okay. So now, a 5-year-old child died from severe septic shock, disseminated intravascular coagulation, cardiac insufficiency following a sudden onset of fever, severe headache, vomiting. The causative agent, a gram-negative Diplococcus, was isolated from blood and CSF, or Thyromartin agar, and was identified in CSF. What is the most important virulence factor involved in disease production by the etiologic agent in this case? So here, even if you didn't know what this bacteria is referring to, what this bacteria is, you know it's gram-negative. You know it caused septic shock and fever and DIC. Which thing causes septic? Yes. Yes. So it's not the polysaccharide. Yes, the polysaccharide capsule helps this bacteria, which is, what bacteria is this? Yes, Neisseria meningitidis. So this bacteria is Neisseria meningitidis. However, yes, the capsule is the one that made it go to the brain and cause meningitis. But the most important thing that caused it to have septic shock, DIC, fever, and all of that is the lipopolysaccharides, which contains lipid A, which is the endotoxin. Okay, so it's important because they said which one is the most important in this case. In this case, the severe thing was the septic shock. He didn't die from meningitis. He died from the septic shock and the DIC. Okay. So the answer here is lipopolysaccharides. Okay. The following picture is an example of a bacteria with which type of flagella or arrangement of flagella? Yes, peritrichus. Amphi is on two sides, Lopho all on one side, and mono is only one per one side. Okay. Clostridium spores are resistant to all except, yes, perfect, autoclave. So it can resist harsh conditions except an autoclave. And speaking of an autoclaving or autoclave, what is it? D. Okay, yes. So basically it's 121 degrees Celsius for 15 to 20 minutes under steam pressure. Okay. Okay. Is this my last question or not? Okay. C. Diphtheria toxin works by the toxins. They work by, yes, intercellular with two subunits. So A and B. Which one binds and which one damages the cell? B binds. Yeah. No, not A binds. Remember that B. Okay. So B binds. A attacks. Okay. So B binds the receptor and then endocytosis and then yeah. Okay. So which one attaches to TCR and MHC2 receptors? Superantigen, Staph aureus. Activating macrophages and complements is by endotoxins. Pore formation is another one, which ones, Shigella and they always come together. They even have the same agar to differentiate between them. Yes, Salmonella and Shigella. And lytic enzyme, pneumonia, pneumolysin. Yes, okay, perfect. So that's the end of my lecture. Thank you so much. If you guys have any questions, you can ask me. And I would really appreciate if you can fill in this feedback so that I can improve myself in the upcoming thing. And yeah, if you guys have any questions, B is endotoxin, yes. It basically causes inflammation, fever, cytokine release, TNF alpha, interleukin 1, so yeah. And yeah. Any other question? Yes, guys, please fill the form so we can improve ourselves. Really appreciate that. No problem. And good luck with your exams. If you guys have any questions, you can always send me a text message or an email. Text message is preferable. But yeah, even if not related to this lecture. Yeah.